In this video, we're going to talk about cell compartmentalization and why that's important in biochemistry. Once again, most of our images and references will be connected to our Cognitive Higher Level Biology textbook. So first of all, what is the importance for compartments? Well, compartments actually allow cells to function much more efficiently than if there were no compartments. Just like a house has different rooms for different purposes, cells have different organelles that do specific jobs. And the functions of these organelles are due to their biochemical structure. If you remember from your grade 11 biology course, you took some time talking about prokaryotic cells and eukaryotic cells. Both of these cells have organelles, but eukaryotic cells have membrane-bound organelles, which allows for this compartmentalization we've been talking about. So that helps that you separate different cell processes and cell reactions. And when we learn our metabolic processes unit, we'll see where this compartmentalization is extremely important. Before proceeding with the rest of the video, you should go to your cognitive textbook and do interactive 1 in the B2.2.1 section and have a look at all the different organelles. This would be within an animal cell. And then identify what their function is and decide whether each is an organelle or not. So take some time to do that. Within that same section, take some time to do the second interactive, and that one has to do with the plant cell. And again, identify whether each of those structures is an organelle or not. And remind yourself of the function of each of the structures. So the following structures are not organelles because they aren't membrane bound and they're not involved in metabolic processes. So those two properties are important for identifying whether something is an organelle. So the cytoskeleton is not an organelle because it's the protein network that helps give the cell its shape, but it isn't membrane bound and it's not involved in a metabolic process. The cell wall isn't an organelle either, even though you may have learned it to be one, because it's mainly made of cellulose, which is a carbohydrate, and it provides rigid support of plant cells. So again, it's not membrane bound. And then finally, the cytoplasm, even though you would have learned in younger grades that it was an organelle, it's not. It's the fluid matrix that makes up the inner part of a cell. However, it is involved in many metabolic processes because it's where those processes take place. And it also holds all the other organelles. Now, having said all of that, ribosomes are considered to be organelles, even though they aren't bound by membranes. Their job is to build proteins, so they are involved in metabolic processes. And that's what lets them be classified as organelles. They're also found often closely associated to the endoplasmic reticulum, which is membrane bound. So we're going to take some time to look at the nucleus and a couple of other structures, but first of all, the nucleus. So it's important to keep all your nuclear material separate from the cytoplasm to prevent the nuclear material from being digested by enzymes that might be found in the cytoplasm. The cytoplasm can send signals to the nucleus to control gene expression. And the nuclear membrane does have pores within it to allow materials to move in and out. And you can see those in the diagram at the right from our textbook. So you can see there's a nuclear pore here and here and here. So the transcripts that are produced during transcription can leave. Other things can go within and so on. The nucleus can also form vesicles or vesicles to package materials to be able to leave the cell. And in our cognitive textbook, in the section that we've been exploring, B2.2, you could actually see an animation where materials are moving through the nuclear pores. If you remember your steps of mitosis and meiosis, you'll remember that during prophase of both processes, the nuclear membrane dissolves. After all of the steps of mitosis or meiosis have occurred, the nuclear membrane has to reform, and the way it does it is it forms vesicles around that are from the endoplasmic reticulum, and they form around each chromosome. Each of those chromosomes that are wrapped in vesicles all join together and then form the nuclear membrane, and all of the chromosomes are contained inside that membrane, forming the nucleus. The Golgi apparatus, its job is to package up proteins. So ribosomes on the rough endoplasmic reticulum build proteins, and the rough endoplasmic reticulum modify those proteins and then transport them to the Golgi apparatus for packaging. A vesicle will break off of the rough endoplasmic reticulum, as you can see here, and then it will carry the protein to the Golgi apparatus, where it will bind to the cis compartment, right here. The cis compartment transfers the protein to the medial compartment, where it will be further modified. And if the protein is to be transported out of the cell, it'll actually be passed to the trans compartment and then off to another cell. So the cis is the one that's closest to the endoplasmic reticulum. The medial compartments are the middle part, and then the trans compartments are the ones furthest away from the endoplasmic reticulum. So vesicles or vesicles are membrane-bound structures that are involved in transport or other jobs. So there's a variety of them. The first one is a transport vesicle or vesicle. They move things from one part of the cell to another. So just kind of like a taxi or a shuttle or a bus. Secretory vesicles send things like hormones, neurotransmitters, and other materials out of the cell. So they secrete out of the cell. Transport is within. Secretory is without or outside of. Lysosomes are special vesicles that contain hydrolytic enzymes to break things down. So they'll break down waste and old organelles and other toxins and materials. And then finally, peroxisomes are like lysosomes, but its enzymes have a more detoxifying role. So they detoxify things that don't belong in the cell. It could be harmful. Finally, clathrin is an important part of forming vesicles, but scientists still don't know exactly how clathrin works. But this diagram and my explanation will do our best to help explain based on our knowledge so far. So it works with the cytoskeleton and cell proteins to form a vesicle. It forms, first of all, a clathrin pit, and then a coat around the part of the membrane that is forming a vesicle. And then eventually, once the vesicle is formed, it will break down again. So you end up with, say you're wanting to wrap up this virus. So you end up with a clathrin-coated pit, so the clathrin molecules come along the outside. Then you end up with a clathrin-coated vesicle, and then you fuse that vesicle with the lysosome, and then it breaks everything down. And then that clathrin cage or coat breaks apart when you don't need it anymore.